Hello students, the topic we'll be seeing today is gingival sulcus and DEJ, dentino-enamel junction. Under this, we'll be covering anatomical parts of gingiva, gingival sulcus, sulcular epithelium, significance of gingival sulcus, gingival crevicular fluid, GCF, dentino-enamel junction development, dentino-enamel junction structure, significance of dentino-enamel junction, the dentin at DEJ, the enamel at DEJ, and structures related to DEJ. Moving on to the anatomical parts of gingiva, it consists of gingival sulcus. This is a gingival sulcus. The gap between the tooth enamel and the gingiva, marginal gingiva, is the gingival sulcus. It is very narrow, so it is not seen usually. It can be felt up to 2 to 3 mm with the periodontal probe in a normal healthy gingiva. That's gingival sulcus. Then the free gingiva, that is from the tip of the marginal gingiva till the free gingival groove is the free gingiva. Next comes the attached gingiva. From the free gingival groove to the mucogingival junction is the attached gingiva. Now the name comes as free gingiva and attached gingiva because attached gingiva is laid on the alveolar bone. If you can see, this area is like stuck to the alveolar bone, and here no bone support is there. So this area is free, hence free gingiva. So gingival sulcus, free gingiva, and attached gingiva. And then interdental gingiva. Gingiva present between any two teeth is interdental gingiva. Gingival sulcus, also known as the gingival crevice. It is a V-shaped notch. V-shaped notch. The gingival sulcus is the space between the tooth and the surface of the sulcular epithelium. This is the sulcular epithelium. This is the tooth. Space between them is the gingival sulcus. Boundaries of the gingival sulcus. Inner boundary is the tooth surface, enamel surface. Outer boundary is the sulcular epithelium. Base of the sulcus is the junctional epithelium. Sulcular epithelium. This is the sulcular epithelium in purple color. It is a non-keratinized structure. Very important one word. Sulcular epithelium is non-keratinized. Sulcular epithelium acts as a semi-permeable membrane through which fluids from the gingiva seep into the gingival sulcus. Cytokeratins of the sulcular epithelium are cytokeratin 4, 5, 6, 13, 14, 16, and 19. Significance of gingival sulcus is anatomy of the gingival sulcus is ideal for bacterial growth because it is resistant to the washing and cleansing action of the saliva. In response to dental plaque, neutrophils migrate into the gingival sulcus. Clinical determination of the depth of gingival sulcus is an important diagnostic parameter in periodontitis. Gingival crevicular fluid, or the GCF. GCF is an exudative fluid, important one word, also known as sulcular fluid or gingival fluid. GCF provides a rich source of nutrients to microorganisms. Predominant type of cells in the GCF are polymorphonuclear leukocytes. Predominant immunoglobulin in the GCF is IgG, important one word. Functions of GCF are: GCF contains plasma proteins that help in adhesion. It possesses antimicrobial properties, exert antimicrobial activity, and the glucose level in GCF is 3 to 4 times greater than that in serum. Important one word. GCF is increased in 3 cases. Number 1, in gingivitis. Number 2, periodontal pocket formation. And number 3, smoking. Now an MCQ here is drug which reaches the maximum concentration in GCF is tetracycline. Development of dentino-enamel junction. The junction between the dentin and the enamel is the dentino-enamel junction. Development of dentino-enamel junction. A structure known as the basal lamina separates the inner enamel epithelium from the odontoblast. During odontogenesis, that is tooth formation, upon contact of enamel matrix with dentin, this basal lamina disintegrates and begins to mineralize to form the dentino-enamel junction. So from this, we can understand that the basal lamina is the precursor for dentino-enamel junction. Structure of DEJ. Dentin is here, enamel is here, and you can see a scalloped line. That is the DEJ. Structure of DEJ is scalloped. The convexities of the scallop face the dentin and the concavities face the enamel. DEJ is less mineralized when compared to enamel and dentin. DEJ assures the firm hold of the enamel cap onto the dentin. Significance of DEJ. The DEJ is a complex critical structure uniting two dissimilar calcified tissues, that is enamel and dentin, and they act to prevent the propagation of cracks from enamel to dentin. Spread of caries at the DEJ occurs in lateral direction. One word to note. Pit and fissure caries affect the DEJ to a greater extent than smooth surface caries. This is again a one word. The dentin at dentino-enamel junction. In the DEJ, convexities of the scallop face the dentin. A band of predentin is present at the DEJ. Dentinal tubules are further apart at the DEJ and much closer at the pulpal surface. This is an important one word. At the DEJ, dentinal tubules are further apart. Osmotic pressure is less at the DEJ rather than the pulpal area. Branching of dentinal tubules is noted at the DEJ. Enamel at the DEJ. In the dentino-enamel junction, concavities of the scallop face the enamel. Enamel rods run perpendicular to DEJ. One word to note. Transitory structures like enamel spindles, enamel tufts, enamel lamellae are all noticed in the DEJ. One word to note. Transitory structures of enamel. These are all seen in the cap stage students, cap stage of odontogenesis. They are noticed at the DEJ. Diameter of enamel rods is smaller at the DEJ and it increases towards the surface. Enamel rods are more densely packed at the DEJ when compared to the outer surface. At the DEJ, the enamel rods are 44,000 per square millimeter. At the surface, it is just 40,000 per square millimeter. Now, if you can see, the enamel rod has a head and a tail. It is a keyhole shaped structure. This is the dentino-enamel junction, this line. The tail of the enamel rod is always towards the dentino-enamel junction and the head is towards the outer surface. Structures related to the DEJ: enamel tuft, enamel lamellae, and enamel spindle. If you can see in this diagram, this is the dentino-enamel junction, enamel and dentin. Enamel spindle comes from the dentin and projects into the enamel. Enamel lamellae is here, and enamel tufts are grass-like structures arising at the DEJ. We'll see about each of them in detail. First, enamel tuft resemble a tuft of grass. They arise from the dentino-enamel junction, reach to 1/3 to 1/5 of enamel thickness, consist of enamel rods and interprismatic substances, extend in the direction of the long axis of the ground. Enamel lamellae. They are thin and leaf-like. They develop in planes of tension. They extend in longitudinal and radical direction. This is the enamel lamellae. They develop in planes of tension. They extend in longitudinal and radical direction. There are 3 types of enamel lamellae. This is a very, very important one word, students. Please pay attention. Type A, type B, and type C. Type A consistency is poorly calcified enamel rods. Type B consists of degenerated cells. And type C has organic matrix from the saliva. Type A enamel lamellae and type B enamel lamellae are found in unerupted tooth, while type C is found in erupted tooth. Type A is restricted to the enamel. Type B reaches into the enamel, and type C reaches into the dentin. So you can see in this image, enamel, dentino-enamel junction, and the dentin. So type C reaches into the dentin. Type B reaches the enamel, and type A is restricted within the enamel. Type A and B are less common, and type C is more common. Moving on to enamel spindles. Enamel spindles are formed by the extension of odontoblastic processes that cross the DEJ and extend into the enamel before calcification. They are found in the cuspal region. Enamel spindles, odontoblastic processes. This is the enamel in this image, and this is the dentin. You can see odontoblastic processes that extend into the enamel, and their extension happens before calcification can occur. In this diagram, you can appreciate it better. Enamel spindles, extension of odontoblastic processes into the enamel before calcification. Enamel tuft is represented better here with this image. Enamel spindle. That's all for this class, students. Moving on to MCQs. Cytokeratins of the sulcular epithelium are all except. Answer is option C. Except for option C, all other cytokeratins are present in the sulcular epithelium. If you go back, kindly refer to what all cytokeratins are present in the sulcular epithelium. Question 2. Precursor of DEJ during odontogenesis is? Answer, basal lamina. So the DEJ develops from basal lamina. That's all, students. Thank you.